Hello guys, good evening. Today we are going to discuss about cholesterol biosynthesis. Why it is so important to learn about cholesterol? What is the need? Because cholesterol is the precursor for the synthesis of steroid hormones, myelin sheath, and it is a precursor for synthesis of bile acids, bile pigments, bile salts. Okay, so it is also required for synthesis of lipoproteins like HDL, LDL. So let's get started. So it is very important to learn cholesterol because it is an LAQ question in the last year also. Okay, when we written our exams, it is our LAQ question. They given a case and they are asked about the cholesterol biosynthesis in one of those 5 questions. Okay, so let's get started guys. So we already knew how acetyl CoA is formed from beta oxidation. Okay, this acetyl CoA will be combined with another more acetyl CoA. This step is known as condensation, presence of acetoacetyl CoA synthase, and we are discussing condensation, so removal of smaller molecules like CoA. Okay, then what will be our product? The product is acetoacetyl CoA. This is a 4 carbon compound since it is formed from 2 molecules of acetyl CoA. So this acetoacetyl CoA will be again combining with acetyl CoA in the presence of HMG CoA synthase. So obviously our product is HMG CoA, which is known as beta hydroxy beta methyl glutaryl CoA, and our third step is reduction. Presence of HMG CoA reductase, we are getting mevalonate, which is 6 carbon compound as that of HMG CoA. And fourth step is phosphorylation. Presence of kinase, ATP is converting into ADP, and in the fifth position phosphate is added. Okay, so we are getting 5 phosphomevalonate, which is a 6 carbon compound, and one more phosphorylation which is occurring so in terms of kinase, wherever the kinase is, there is ATP. So we are adding ATP, means ATP is converting into ADP, and one inorganic phosphate which is removed, that inorganic phosphate which is added at the again fifth position. So 5 pyrophosphomevalonate we are getting. Again we are adding ATP. This time third carbon we are adding phosphate, that inorganic phosphate. So we are getting 3 phospho 5 pyrophosphomevalonate. Okay, here up to now there is no carbons in 6 carbon compounds only. These are all mevalonate, 5 phosphomevalonate, 3 phospho 5 pyrophosphomevalonate. These are all 6 carbon compounds only. Okay. So now what is happening? Now decarboxylation is happening. So one molecule of carbon dioxide which is removed, means out of 6 carbons, 1 carbon is removed in the form of CO2. Then we are getting 5 carbon compound which is isopentenyl, as it is 5 carbon compound. Inyl means, in means one double bond is there. So pyrophosphate we are getting. Then in the presence of isomerase, some rearrangement will occur in the structure of isopentenyl pyrophosphate. So we are getting dimethyl allyl pyrophosphate, which is also a 5 carbon compound. Then we are means we are adding it with isopentenyl pyrophosphate. This is which is 5 carbon compound. Then what will be the product? Product will be 10 carbon geranyl pyrophosphate. Okay, again we are adding isopentenyl pyrophosphate to the geranyl pyrophosphate. Then we are getting farnesyl pyrophosphate, which is a 15 carbon compound. So obviously general geranyl pyrophosphate which is 10 carbons, and we are adding isopentenyl pyrophosphate which is 5 carbon. So we are getting 15 carbon. Okay, done. We'll continue now. So this farnesyl pyrophosphate, we are again adding one more farnesyl pyrophosphate which is 15 carbon. 15 plus 15, 30. So 30 carbon squalene we are getting. So in presence of squalene epoxidase, we are getting squalene epoxide as a product. Then cyclization occur. So then squalene epoxide will be converting into cyclical form lanosterol. Lanosterol will be converting into zymosterol by removal of 3 carbons, and we are getting 27 carbons zymosterol, and zymosterol will be rearranged into desmosterol. Desmosterol finally leads to the production of cholesterol. Okay, this is a cholesterol structure, guys. They will keep it as Potter, and they will ask about the nucleus, what this nucleus which is called as, and they will even ask where the position of double bond is situated, like that. From the fifth to sixth portion there is a one double bond, and third portion there is hydroxyl group. Okay, so how every cycle, if we describe any metabolism, the regulation which is very important, regulation plays a key role, and whether they ask or not, we have to write about the regulation. Okay, so statins are competitive inhibitors. Okay, like levostatin, rosuvastatin. So many statins are there. These statins will inhibit the HMG-CoA reductase, so that it is a rate limiting step, right? What is rate limiting step? Rate limiting step means it is the slowest step which decides the order of whole reaction, and if it won't happens, the whole metabolism doesn't occur. It is very important step, and it decides the metabolism as the whole. So these statins are inhibiting the rate limiting enzyme HMG-CoA reductase. Okay, then glucagon, cortisone, these are also inhibiting the HMG-CoA reductase. Okay, then cholesterol itself in the excess in feedback negative feedback mechanism itself, it is inhibiting its synthesis. And even insulin, thyroxine, mevalonate, these have the positive role in HMG CoA reductase, means they will promote the cholesterol biosynthesis. Okay, immediately after eating, which hormone is high in our blood? Insulin, obviously. Insulin will help in cholesterol biosynthesis. Okay, and then how we will regulate the HMG CoA reductase, means by means of this we have to remember this covalent modification. And as you guys read the enzymes chapter, there we will study about covalent modification. Covalent modification plays a key role. Once go on study. So HMG-CoA, with the help of HMG-CoA, we are getting cholesterol in presence of HMG-CoA reductase, which is a rate limiting enzyme, and it is active in the dephosphorylated form. So reductase kinase, if it is present, HMG-CoA will undergoes phosphorylation and becomes inactive. So to become active, we want a protein phosphatase enzyme. So insulin will promote the protein phosphatase and activate protein phosphatase. So HMG-CoA reductase will be in the active form, and glucagon will stimulate the cyclic AMP formation. Actually, glucagon, what will happen? Glucagon will activate adenyl cyclase, and it will convert ATP into cyclic AMP. The cyclic AMP will cause a phosphorylation of the protein kinase. So protein kinase from inactivated form, it will become activated, and this protein kinase which is activated, it will donate the phosphate to the dephosphorylated enzyme, and make the enzyme phosphorylated. But in presence of insulin, the cyclic AMP will be immediately converting into the 5' AMP, so that the protein kinase remains in inactive state, and protein phosphatase will be the active form, and this protein phosphatase will convert the inactive HMG-CoA reductase into the activated HMG-CoA reductase. Okay, this is regulation of HMG-CoA reductase. Then this dietary cholesterol, what is the fate of cholesterol? Means this dietary cholesterol will be converting into the chylomicron remnants. Okay, chylomicrons are majorly found in the intestine, and these remnants are formed from the cholesterol. Okay, protein coated fat globules, and this cholesterol from extrahepatic tissues. will be getting converted into HDL and is transferring into the liver, which is good cholesterol. Okay, and de novo biosynthesis in the liver, cholesterol pool will be occurring in the liver. This cholesterol pool will be helpful for the secretion of the VLDL, free cholesterol, which is secreted in the form of bile, and conversion of bile acids or bile salts. Okay, this is all about cholesterol. Okay, cholesterol biosynthesis. Hope you enjoyed this lecture, and if you have any doubts you can comment, ask, and thank you for carefully listening to me and patiently being with me all this time of this 11 minutes and listening to this topic carefully. Thank you guys, thank you so much for listening to me patiently. Thank you.